So our first speaker this session is Alessio Crippa, and his talk is a flexible dashboard for monitoring platform trial. So let me go full screen. And Alessio, are you here?
Yes.
Okay.
Are you able to share the screen with the remaining presenters? Is it okay?
I think I am sharing my screen. Do you see it?
Yes, I see it.
Okay, there you go. You have 3 minutes.
Okay, hello everybody. My name is Alessio, and I'm a postdoctoral research in Stockholm at the Karolinska Institutet. Currently, I'm working on a ProBio study, which is a platform clinical trial in which we're trying to implement genetic biomarkers, so the mutation of the tumor, in order to decide which treatment best fits one patient, and this is for advanced prostate cancer patient. And the idea of the platform trial is that the number of treatment will change. Some treatment will be stopped earlier because of graduation, meaning that they perform really good, or we are going to stop them because they are performing worse than expected. And we're also changing some other elements, such as randomization probability, such a way that more patients will benefit for the most promising treatments. And this requires that we keep monitoring the study progress daily, or even every single hour. And that's what motivated this poster. This is a way how I show how to fetch the data constantly by writing an API interface in R, how I developed 2 scripts for cleaning the data, manipulating the data, and then analyzing them in order to produce meaningful table and results also from more complicated model. And then I store those results and I present them in a dynamic report, which is accessible with different level of confidentiality to different people involved in the trial. So the end results of this poster is just a shell script which on a cron tab job scheduled on a server run every midnight those 4 synchronized scripts and generate these dynamic reports with different results, which may be of help for the people involved in the trial. Among others, the sponsor of the trial to see the accrual rate, but also for the statistician, as they may evaluate the effectiveness of the different treatment, but also the data monitoring board, which will advise the sponsor to make some changes throughout the trial depending on the data presented, as well as for the safety of the patient enrolled in the trial. So if you're interested in the poster, just write me an email, or I will try to be in the session there if you want to speak more about that. I don't know if I stick with the 3 minutes or more, but I'm here if you have any more questions. Thank you.
Yeah, you're perfectly on time. 5 seconds left. So thank you, Alessio.
Thank you.
Check out his room if you want to know more about his project. So next up is Andreella, and she will be talking about the PRDA package, announcing statistical inference via prospective and retrospective design analysis.
Hi everyone, another time. Okay, so I will present a project from of the Psychostat group from the University of Padua that I'm a member. This project is about the PRDA package. That is a package I think very useful for the researcher that I wish to make design analysis. This package permits to perform then to evaluate the inferential risk of your experiment, but it permits not only to compute the most famous power analysis, but also to compute the type S error and the type M error from the definition of Gell-Mann and Curlin. Briefly, the type S error is the probability to detect a significant effect size in the wrong direction, while the type M error is the value of the amount of overestimation of the detected significant effect size. And also, the PRDA package permits to perform a prospective and retrospective design analysis, so before and after data collection. The output is mainly the same, but in the case of prospective design analysis, we have also the optimal sample size. We developed this design analysis considering mainly two effect size very used in the psychological field, the Cohen's-d and the Pearson correlation. But we wanted to expand this package, consider other effect size, and also using a Bayesian approach. So I don't know if I use all the 3 minutes, but if you are interested, I will wait for you in the speaker room. Thank you.
Perfect. Thank you, Angela.
Thank you.
Okay, so next up is Dario Righelli, and he will present EasyReporting, a Bioconductor package for reproducible research implementation.
Hi. So, hi. Okay, I see the slide. Okay, so this project is a package, mostly a class for helping to implement reproducible research inside other projects, packages or software in general. And this class can be thought as a representation of the R Markdown file, where the attributes store the report characteristics, and the methods has these can be used for the manipulation of the attributes and also for the code chunk insertions. You can also add comments, titles, and some other functionalities. You can construct a code chunk step by step. So by opening the chunk, inserting the variables that you want to trace or the function that you want to track, and then to close the code chunk. Otherwise, there are some other functions that are able to. It's one call function that you can do to construct the code chunk and also to add personalized comments to that code chunk. Also add the title and whatever. The class automatically constructs the R Markdown file, and then by using the method compile, it constructs the HTML files. Also, if you are interested, I prepared a live demo. So if you are interested, you can come in my room, personal room, so we can discuss and I can show you other functionalities about this class. That's all for me. Thank you.
We're going to move on. The next speaker is Federico Agostinis, who will present NewWave, a scalable R package for the dimensionality reduction of single-cell RNA-seq.
Hello, everyone. And I'm going to speak about NewWave. It is a development of the existing R package ZimWave, designed for dimensionality reduction and batch effect removal. It is formed by 4 main steps: the initialization, the optimization of the dispersion parameters, the optimization of the cell-related parameters that can be done cell by cell, so it is theoretically massively parallelizable, and the optimization of the genes-related parameters that is done gene by gene, so it could be theoretically massively parallelizable. It has two main limitations: the memory consumption, due to the high number of parameters in the zero-inflated negative binomial model, and when it is used in a parallel way, it has an greatly increases of the memory consumption, linearly proportional to the number of cores. And the second limitation is the time consumption due to the high number of parameters, and it cannot be used with a lot of cores for memory saturation. So I created NewWave that assumes a negative binomial distribution. And for the parallelization, I used the PSOC cluster combined with a shared object package that creates a physically shared memory between cores at allow in-place operation. I used the delayed array Bioconductor package for work with the HDF5 files. And this is the comparison between the RAM consumption of the tube model. To reduce the time consumption, I implemented some mini-batch approaches, but also the reduction in memory consumption could reduce a lot the time consumption. And I'm now benchmarking the time consumption and the performance of this model. And in the QA session, I will make you see some of this benchmark. And I will improve the delayed array framework to optimize the access to the block. And thank you.
Federico, perfectly on time. And now we can move to the next speaker, it's Patricia Ryser-Welch, who is talking about integrating professional software engineering practices in medical research software. Patricia, you have 3 minutes. Probably joining. Sometimes Hopin is a little slow at allowing you to share your audio and video.
Oh, hello. Hi.
Here you are. All right.
Yeah, I'm here. Yeah, because I'm in United Kingdom. You know, this part where we live in EU. That's fine. There you go. They cut the internet. Hi, my name is Patricia Ryser-Welch, and I'm presenting some work on behalf of the University of Newcastle, and especially the project called Data Shield. What we're doing something much different from the previous 3 presenters. We are a non-disclosive federated analysis, and what we do, we work especially in medical research software, but our software can adapt to many other types of data. The reason why we work with we basically engage with medical research software, it's because a lot of people in medical science appear to keep the data in spreadsheets, or the strict data governance tend to keep the data basically where they live. So it's very hard to have access to them. So that is why there is some trusted research environment which allow certain people, certain researchers to have access to some data with some strict data governance agreement. And what we did put some non-disclosive and federated analysis on top of this trusted environment, so that we added another layer of anonymization. But the user we use, they can affect life and death sometimes through their analysis, because they would inform decisions that can be made for example, on medications or type of living, and its current week it can as well aspect of different communities. So what we did, we brought some testing to convince user for accuracy and mathematical correctness, as we provide a lot of statistical functions, and we want to prove to our users that what we're doing is correct. And what we have done to prove even further, I need to adapt to new technologies with a continuous integration and test using industry standards. R is keep improving, operating system keep improving, library change, and like the CRAN person said this morning, say it's quite hard to keep up. So we tend to put Azure to the limit of its capability. We're quite proud of that. And we test everything on different operating system, different new version, older versions, such as our software, Data Shield, remain of good quality. So if you want to know a bit more how you can build trust and enable engagement using extensive testing framework and online versioning technologies, and using this trusted research environment, come and see you in my room. I will definitely be happy. And tomorrow we've got a workshop about non-disclosive data analysis. So come and join me then as well. But I look forward to see you later on.
All right. Thanks so much, Patricia. And I'm really impressed by how well the speakers are keeping the 3 minutes mark. They're making my work really easy. All right.
We've got timers, that's why.
All right, so check out Patricia's room if you want to know more about her project. Next up is Tobias Schieferdecker, who is going to be talking about dealing with changing administrative boundaries, the case of Swiss municipalities.
Thanks, Davide, and hello, everyone. Just gonna turn off my video. Okay. To introduce my topic, we all know that worldwide analyses are performed on the different administrative levels of countries. However, these units can change over time. The units can merge together in the hope to save on administrative costs. They are renamed, they sometimes split up, etc. This makes creating consistent and meaningful time series a challenge. We are proposing a solution in form of an R package for this problem, for the specific use case of Switzerland. This may sound a little bit niche maybe, but the idea can be generalized to any other country as well, depending on how their statistical offices are providing the information about their changing administrative boundaries. The Swiss Federal Statistical Office provides this information in a compact table with 1 row per mutation, which is great, but not in all cases easy to use. Our approach is to blow up this table in such a way that it is possible to just join the table containing your data to this mapping table. By doing this, you are mapping all those municipalities that were separated from the municipality of interest in the beginning of your time series to this municipality in case they have merged before the end of the time series. On this slide here, we are showing an example of the problem and of our suggested solution. We are looking at time series of the population of a subset of municipalities in the canton of Zurich. On the left plot, you see the original data. Everything on it is correct, but some of the time series are ending early and others show sudden increases. With a simple join operation of the population data with the appropriate mapping table, you get an impression of how the past would have looked had those municipalities always been united. So Thomas and I are really happy to have a chat with you if you are interested. So yeah, please drop by if you want to hear more about this. Thank you.
Thank you, Tobias. And I think we are at the end of our introductory session. And then, like as I said at the beginning, you will find in the Sessions tab at the left of your screen the rooms, 1 for each poster with the name of the presenter. And I hope you're going to check out those rooms and ask a lot of questions to our fantastic presenters. And a big round of virtual applause to all of the people who presented the poster. And thank you.